In this video, we're going to complete our coverage of the visual system, and we begin by examining the first layer of cells that come in contact with the electromagnetic energy coming in into the eyes towards the back of the eyeball, and those are the photoreceptors. And so the photoreceptors are those specialized cells, as you can see drawn here on this slide, and number at approximately 100 million per retina, and that's in each eye. And these cells, these very odd-looking cells, are actually neurons. Of the photoreceptors, the rods make up the vast majority of these at about 90 million, and the cones account for approximately 5 million or so of these cells. Now, I'll begin here by examining rods, and kind of a handy way to remember the function of the rod photoreceptor is to think about a lightning rod. And so in this picture here, we see a lightning rod capturing lightning at night in the evening. So rods are the photoreceptor cells that are particularly tuned in operating under low light conditions. And all rod cells, and there are approximately 90 million of them, all have the same photopigment. And having the same photopigment, which is the molecule that responds to the electromagnetic signal coming in, does not allow these cells or limits these cells' ability to differentiate between different colors. So these are cells that cannot do that. But rod, think about the lightning rod, lightning, very luminous at night. And so rods help us to track light in low light conditions. Opposite to that are cones. And handy way to remember what the function of cone cells is that I like to think about ice cream cones that people typically eat during the day. And so for cone cells to be activated, I think about eating an ice cream cone during the day when there's lots of sunlight out. And so for these cells to be activated, it requires brighter light. Now, as you can see on the right-hand picture here, we have many more colors that are quite uh, uh, visible as opposed to the variety of colors visible at night. This is certainly illuminated by all this other artificial and natural light, but under low light conditions, the ability to differentiate colors is very low as compared to daylight. And that's because cones have three different pigments, are called photopigments. And as a result, the cone cells have the ability to signal the different wavelengths of light. So think back to the visible light spectrum um, between 400 and 700 nanometers with the different colors, and the region in between with that transition of the colors of those wavelengths. Some more specific details on the photoreceptors. What photoreceptors, although rods and cones are certainly quite a bit different, they do have some similar properties. And the both have what's called an outer segment, and this is the segment that is most posterior, that is positioned towards the pigment epithelium at the very back of the eyeball, back of the retina. And the outer segment is the portion of the cell, both the cones and rods, that contains the photopigment. As we move towards the inner part of the eye, from the most peripheral part of the retina, we have the inner segment. And one of the functions of the inner segment is that it's the part that actually manufactures the photopigment in these cells that will migrate to the outer segment, and it makes those pigments particularly sensitive to the wavelength of light. And so once these cells have become activated through these molecules, that will trigger the synaptic terminal. And you can see the synaptic terminal here interacting with what are called bipolar cells. Uh, this is the site where neurotransmitter is released. So we have a chemical synapse where we have a small gap of extracellular fluid between the two different cells, and the proximal cell, the presynaptic cell being the rod or the cone, will release that chemical compound into the extracellular fluid where it will migrate through and form chemical bonds with proteins on the postsynaptic membrane, discharging the activity of the, or creating discharge activity of the bipolar cell. Now, in terms of photoreceptor distribution, it's not constant here. So the cones, which are the photoreceptors that are particularly sensitive to different wavelengths and can give rise to eventually the perception of different colors during the daytime, are quite concentrated at the fovea, that very small indentation at the back of the eyeball. And you can see here in this bottom graph along the x-axis, the 0 refers to the center of the fovea, so the distance being 0 from the center. And as we move towards what's called the nasal retina, the part of the retina closer to the nose, and the opposite direction towards the temporal bone of the retina. So more lateral position, more medial position of the retina. We see in the red line here, or the red curve, representing the density of the cones. And so that's illustrated here on the y-axis as thousands per square millimeter. It's highest right here near the middle of the fovea, but then drops off quite quickly as we move towards the peripheral part of the retina. And the concentration of rods, the rod photoreceptors, which are the ones that are active under conditions of dim light, drop to 0. So there are no rod receptors, photoreceptors at the center of the fovea, but as we move away from that middle part of that very concentrated indentation of the retina, we see that the population of the rod photoreceptors increases very quickly and becomes non-existent for both rods and cones at the optic disc. So the optic disc is slightly medial to the fovea, closer to the medial part of the head towards the nasal region as opposed to the temporal bone. And that's the blind spot. So there are no photoreceptors at the blind spot because there's no tissue at the blind spot other than the blood vessels and the optic nerve moving through it. And so what we see here is that the cone concentration towards the peripheral part of the retina on the nasal side and the temporal side drops very, very low. And although the rod concentration is considerably higher at the periphery of the retina, it also drops off consistently towards the most medial or the most lateral part, the most peripheral part of the retina, making our ability to distinguish different wavelengths towards the peripheral part of the retina much, much lower than it is in the more medial aspect of the retina. So in terms of the photopigments, those molecules particularly responsible for the detection of different wavelengths of light, the photopigment is a molecule made up of two major components here: opsin, which is a protein, plus a chromophore. And the chromophore is that part of the photopigment that captures, in a sense, the photos, if you will, the electromagnetic energy of the light coming in. And the chromophore is selective in terms of absorbing that light, and it's based on the particular wavelength, and the wavelength is eventually perceived as a different color. One particular type of chromophore is called retinal. And retinal is another word, or is another term for one type of vitamin A. There are three types of vitamin A, but the one type of vitamin A found in the photoreceptors as part of the photopigment in the chromophore is called retinal. Rhodopsin is a pigment that's found particularly in rods. So it's the photopigment that goes by a particular name that since it's found in rods, this is the photopigment that gives rods the ability to detect light in low light conditions. In terms of the cones, which are the ones that are the photoreceptor cells particularly tuned to bright light conditions, each cone will contain one of three different pigments. So although all rod cells have the same type of pigment, the cone cells are tuned to the different wavelengths because each cone will have one pigment, but there are three different pigments across all the cone cells collectively. And each pigment is tuned to short, medium, or long wavelengths. And each wavelength will give rise to the perception of a different color. So, in terms of what photoactivation is, photoactivation are basically photons that are absorbed by the chromophore. So, these photons, you can think about the photons actually taking a photo, right? It captures a picture that is conveyed as wavelengths. And this absorption will generate what's called a hyperpolarization. The hyperpolarization is a change in the resting membrane potential, although we don't go into that in great detail here in this particular video, but the electrical environment inside and outside the cell changes because of the concentration of ions inside and outside the cell. But the hyperpolarization will cause that change in the electrical environment will cause the synaptic vesicles, the synaptic terminals to release vesicles that contain glutamate. And glutamate is an amino acid that functions as a neurotransmitter for the chemical synapse from the presynaptic cell, which is um, the photoreceptor, and the bipolar cell, which is the cell communicating with the photoreceptor. And this one is excitatory. Being excitatory, glutamate will cause an activation of the cell that the photoreceptor interacts with, that we see here on this slide. So in terms of the photoactivation sequence, the photon will lead to a photoreceptor cell activation. And just to reiterate on slide 19, that's a hyperpolarization. So if we take a look at this top image here, this drawing, we have our light coming in from the left that moves through this translucent layer, these translucent layers of cells until it reaches the pigment epithelium interacting with the photoreceptors. And inside the photoreceptors, we have the molecules absorbing it, causing the hyperpolarization that will cause a synaptic release of glutamate to the bipolar cells. So the last layer here, so the bipolar cells interact with the photoreceptors, which will then interact with ganglion cells. So the ganglion cells are the final layer in the transduction pathway, which starts at the most peripheral part of the back of the eye and will work its way back towards the inner part of the eye. But keep in mind, this entirety here is a very thin layer at the back of the eye. That the ganglion cells number in over a million, are estimated to be about 1.25 million per retina, and are made up of two fundamental types of ganglion cells. The first are referred to as P ganglion cells, and the other are referred to as M ganglion cells. And these are cells, as you can see here, will merge with the inner limiting membrane, and the P ganglion cells, that's not differentiated here in this picture, but it's these yellow cells that come in two different varieties. The P ganglion cells are named according to the fact that these cells will project to the LGN, which I'll define here shortly, particularly the parvocellular layer of the LGN. The M ganglion cells will transmit and project to the magnocellular layer of the LGN. And magno means that this is a larger layer of cells in the LGN, parvo referring to a smaller layer of cells in the LGN. And the LGN stands for the lateral geniculate nucleus, which is a cluster of neurons located in the thalamus. And recall from earlier units, the thalamus is a structure in the brain that serves as a relay station. It receives multiple sensory inputs. There's a slight pause as synaptic connections reorganize those sensory inputs and will then project and send bioelectrical information out of the thalamus to the respective regions of the brain involved with the perception of that unique sensory input. And the central visual pathway is the pathway that gives rise to the perception, so the seeing part of vision. And a handy way to remember the pathway here is the saying, never catch the lion running slow, using the first letters of each word, referring to the transmission pathway from initial sensation to final perception. And it starts with the optic nerve. So this is occurring after phototransduction, so that the electromagnetic energy has entered the eye and has caused the activation of the rods and cones, so that the neurotransmitters are released that will cause the final bioelectrical activation of the M and or P ganglion cells. And those will transmit along the optic nerve, which is also referred to as cranial nerve 2. And the cranial nerve 2, this is the nerve made up of sensory fibers only. So this is the nerve that will transduce electrical energy from electromagnetic to the regions of the brain. Chiasm, or the optic chiasm, is a continuation of the optic nerve, but is referred to as that region where there is some crossing of the cranial nerves. Not all will cross, but we'll see that on this picture here shortly. So that's the C of the never catch the lion running slow. The T is the optic tract. So let me just step back here one point here. The optic chiasm having the ability where we actually see a hard wiring of these neurons that will cross over to the other side of the brain, will give rise to binocular vision. So the simultaneous transduction of electromagnetic energy between the right and left eye. The optic chiasm will then merge into what's called the optic tract. The optic tract is a collection of the neurons transmitting this information over to the LGN, the lateral geniculate nucleus, which is part of the thalamus, our very large relay station centered in the brain. From the thalamus, the small delay will then reorganize those neurons and project out through what's called the optic radiation. And the optic radiation, just as the term suggests, will project neurons that will radiate out from a relatively concentrated area in one part of the thalamus and will terminate into a relatively larger region of what's called the striate cortex. And the striate cortex is located in the occipital lobe, and that's where we then will perceive the sensation of electromagnetic photoactivation that started in the retina. So if we take a look at this picture here on the right-hand side, we have the optic nerve, which is collecting the neurons from the M and P ganglion cells. But notice here that as we move towards the optic chiasm, these are all the neurons that are transmitting. You can think about this as being a hardwired system that will transmit to the opposite side of the brain. So the right eye will transmit to the left side of the brain, but will also transmit to the same side of the brain. And we see that with the left eye as well. So we see crossing from the left eye to the right side, and we see that neurons activated, the M and P ganglion cells that give rise to the optic nerve will also be wired to the same side of the brain. And this depends upon originally the light projection into the eyes that hit the retina. So the blue lines here represent light coming in, hitting the right side of the left eye and the right side of the right eye. And we can see that retinal side will stay in terms of the side of the eyeball. So the right side projects to the right brain. Light detected on the left side of the retina for both eyes, left side here, will project to the left side of the brain. And these neurons are collected through the optic tract and will terminate in synapse in the lateral geniculate nucleus of the thalamus, small delay, and then we have the synaptic connections, which will then move through the optic radiation. So these neurons will project to a much wider region into the striate cortex, which is made up of the primary visual area, also referred to as V1, capital letter V1, as well as to what are called association visual areas. Okay, and that ends our final video here on the visual system.